There are structures other than filopodia that cells make that require tight actin bundling. Microvilli, for example, also need structural support and which is provided by actin filaments. In case of microvilli, there's an additional protein which helps organize these actin filaments. The name of that protein is vilin. This protein also has two actin binding sites like filamin that we talked about. So let's look at this microvilli and how vilin helps form these structures. Vilin, along with fibrin, cross-links 20 to 30 tightly bundled actin filaments found in microvilli. I would like to mention, just in case you don't remember, these microvilli are finger-like extensions of plasma membrane on surface of many epithelial cells. For example, a single absorptive epithelial cell in human small intestine has several thousand microvilli on its apical surface. The microvilli are about 0.08 micron wide and 1 micron long, making the absorptive surface of the cell 20 times greater than it would have been without these structures. When vilin is introduced into cultured fibroblasts that don't normally contain vilin and have only few microvilli, the existing microvilli become greatly elongated and also stabilize new ones, and also it results in production of new microvilli. Here's the structure of microvilli. You can see their actin filaments. They are bound, held together tightly. Additionally, we have another molecule here. We have myosin 1 molecule, which is attaching the microfilament or actin filament array to the membrane. The actin filament core of the microvillus is attached to the plasma membrane along its side by these lateral side arms made up of myosin, which has binding sites for filamentous actin on one end and a domain that can bind lipids on the other end. These two types of cross-linkers, one binding actin filament to each other and other binding these filaments to the membrane, seem to be sufficient for the formation of these structures, the microvilli. Now I'm going to talk about another protein which we have talked briefly about, spectrin. Spectrin is a tetramer of two alpha and beta subunits, and the tetramer has two actin binding sites spaced about 200 nanometers apart. I would like to point out that fibrin, the microtubule binding sites on fibrin are 14 nanometer apart, and these sites are 30 nanometer apart for alpha actinin, about twice the distance of fibrin. Spectrin is concentrated just beneath the plasma membrane, where it forms a two-dimensional web which is held together by short actin filaments. That's why we're talking about spectrin, because it can also bind actin filaments, although short little actin filaments which are present on the surface of a membrane. Spectrin links this web to the plasma membrane because it has separate binding sites for peripheral membrane proteins. If you remember, peripheral membrane proteins are the proteins that do not have transmembrane domain and adhere only temporarily to other molecules, which are integral membrane proteins, which are permanently embedded in the plasma membrane. So spectrin links this web of plasma membrane because it has separate binding sites for peripheral membrane proteins, which are themselves positioned near lipid bilayer by these integral membrane proteins, which are permanently embedded in the plasma membrane. Here I have this figure to explain this, to show you how this is done, how cells do it. Here you have transmembrane proteins that have a domain that is embedded in the plasma membrane. This green rod is a short actin filament, and these twisty red things, which are twisting around each other, forming a helical structure, are the spectrin molecules. And if spectrin for some reason is not functional, if we cleave spectrin molecules specifically, it will result in blebbing of membrane, and ultimately it will lead to cell death and degradation ultimately. So it's an important structural element. Now I would like to talk about a protein that forms these web-like structures. That job is done by filaments. They were discovered as the first family of non-muscle actin-binding proteins. They are large cytoplasmic proteins that cross-link cortical actin into dynamic three-dimensional structure. Filament has two actin binding sites with a V-shaped linkage between them. It cross-links actin filament into a network with the filaments oriented almost at right angles to one another, promoting formation of loose and viscous gels by clamping two actin filaments roughly at right angles with each other. I will show you a photograph of how these molecules are holding actin filaments. The actin gels are required for cells to extend thin sheet-like membranous projections called lamellopodia that help cells to crawl on a solid surface. Here you have on the screen, you can see you have actin filaments in red or orangish color, and the green proteins, V-shaped proteins, are the filament dimers, and you can see they are holding these actin filaments roughly at right angle with each other. So this is how filament proteins help link actin filaments, which results in formation of these web-like structures.